7, don't fret. You don't need to know all the details about fret, but do know that it is one of the coolest things that we've talked about yet. So, fret stands for fluorescence or Förster Resonance Energy Transfer. Big fancy way of saying you have two fluorophores, so molecules that can absorb and give off light. And the first fluorophore, it lets off energy that excites the second fluorophore, and then the second fluorophore gives off light. And then you can detect the light that's given off by the second fluorophore when you shine light that excites the first fluorophore, but only if they're close together. And so, because you have this distance dependency, you're able to measure whether their molecules are close together. Are molecules binding? Or is parts of the same molecule, are they close together? So you can measure like conformational dynamics, you can measure binding, all sorts of really cool stuff. We can talk about fluorescent proteins as well as small molecules. So let's do it.
Electrons in a molecule can occupy distinct energy levels, so they can have like a ground state and excited states. I like to think of these as kind of like different floors in an apartment building. The electrons can move between these energy levels, similarly to how you can move between floors in that apartment building, but they can't occupy like the in-between space. So you can't just like levitate in between floors. Now, these like electronic floor plans, these are typically drawn out in these things called Jablonski diagrams, and they show the required energy to get between the different energy levels. These floor plans are going to be different for different molecules and different parts of the same molecule and all of this good thing stuff. And so the amount of energy that's required to go between those floors is going to vary depending on the molecule. Just like how it takes energy to like pull that elevator up floors, but then it takes energy for these electrons to get excited. But then just like you release energy if you were to go and like drop the elevator, cut the cable up when it was at the top and let it just fall down, you get a lot of energy back. Similarly, the electrons, they have to release that energy that they absorbed, that they got excited from. And then they can do this in multiple ways. So sometimes what's going to happen is actually pretty boring. They're going to absorb energy in some form, and then it's just kind of like going to fizzle out. So remember, it has to give back that energy because energy, all that good old physics conservation of energy stuff. So it has to give back that energy. But sometimes it just does this kind of like boring ways with heat and vibration and stuff. We call these non-radiative processes. But it can also do a lot cooler stuff, such as give off light. And when we have a molecule that's giving off light, when we have the electrons falling down and giving back that energy as light, we call that luminescence. We have different terms that we use that depending on where that excitation energy came from. So maybe your elevator went upstairs because you were actually physically cranking a cable, or maybe it went upstairs based on like an electric system. Basically, there are different ways that electrons can get excited that you can give them that perfect amount of energy to take them to the next floor. And one of these ways is through light. And in this case, the excitation energy comes from light, which remember is just little packets of energy traveling in waves. And so in this case, we call it fluorescence. So fluorescence is when you absorb light of one wavelength, your excitation wavelength, and then give it off at a different wavelength, the emission wavelength, which, as we'll talk about, is typically a lower wavelength. Because this isn't totally efficient, you lose a little bit of the energy, and so you're going to give off a lower energy, longer wavelength light, and that'll be your emission. You can also have the energy that's excited you coming from something other than light. So if it comes from a chemical reaction, we call it chemiluminescence. And if that chemical reaction was catalyzed or sped up or mediated by enzymes, we call it bioluminescence. So we'll talk more about that when we get into enzymes. But let's focus, we're going to focus mainly on fluorescence today, which remember is when you have the absorption of light and then you give off light. We'll talk about your plain old fluorescence, but we'll also talk about something called FRET. And FRET is really, really cool. So FRET stands for Förster or Fluorescent Resonance Energy Transfer. Basically, what happens is that if you have two fluorophores nearby one another, If one fluorophore emits light of a wavelength, and remember, just when we're thinking about a wavelength, just think about energy. So if it emits light that has the energy that excites another fluorophore, so another molecule that can absorb light and fluoresce, and if it excites that other fluorophore, then the fluorophore can kind of like steal the energy that the first one would have given off and then give off its own light. So when you shine light that excites the first one, what you're going to see is actually the emission of the second one. And so we'll get much more into detail about what's actually going on. But crucial to this is that it can only happen if the fluorophores are close. So when we're talking about close here, we're talking about like less than 10 nanometers. And remember that an angstrom is 0.1 nanometers. And so basically what this is going to be is about 100 angstrom. But that's like the maximum. So typically what we're dealing with is more like about in the 5 nanometer range. And as we'll see, you can actually calculate distances based on the amount of frets because it is distance dependent. So you can use it as a kind of like spectroscopic ruler to see how close together two molecules are, or to see how close together two parts of the same protein are. So there are really cool things that we can do with fret, and we'll take a look at a lot of examples. I do not want you to get tied up in all of these examples and trying to memorize things. Instead, focus on the concepts and just use the examples as examples to show all of the different types of things that you can do with fret, and start thinking about how you might design experiments to test different things. Similar to fret, but without giving off fluorescence at the end, there's this thing called quench fluorescence. Basically what happens here is that second molecule is going to absorb that energy from the first molecule. It's going to do that whole fret stuff with the first one, but it's not going to actually give off light. Instead, it's a quencher. So it just absorbs that light, but it doesn't give it back as light. So yes, it's going to give it back as like heat and all that boring stuff, but it's not going to give off light. So what happens if you shine light that excites the first fluorophore, you're not going to see any emission if they're close together. But if they go apart, then you can see that emission of the first one. Remember, the quencher doesn't have an emission. And then this way you can kind of detect whether the molecules are interacting, whether they got separated, that sort of thing. So here's an example of a quencher. We're not going to talk too much about quenchers, but this is a common one. It's called Dabsel. And you can see that basically you're only seeing an excitation wavelength, not an emission wavelength. So basically it can absorb this light, but it doesn't give anything back that you can see. But now let's talk more about what the cases in which you shine light at the first thing and you do see light given off. So we're talking about like fluorescence. So going back to like the basic physics of light, remember that it's just these little packets of energy called photons that are traveling in waves. Now, all light is going to travel at the same speed. That's why we have that thing like the speed of light. But that doesn't mean that they all have the same energy or that they all have the same wavelength or frequency. Instead, basically, if you think about a little kid and a grandpa that are kind of like walking together, and the little kid has a lot more energy than the grandpa. So this little kid is kind of like running back and forth and back and forth and back and forth, making these little like zigzags, these like slalom run types things, like those S's that the skiers make. And so the little kid is kind of like running side to side more than the grandpa. And so what's going to happen is that basically it's going to have a shorter wavelength. It'll be doing those up and downs more frequently, so you'll have a higher frequency and a shorter wavelength in order to have all those up and downs, still keep track with grandpa, still keep track with all of that light that has a lower energy. So the energy, remember, is held in those little packets called photons, and it's those photons that different molecules can absorb. So different photons have different amounts of energy. So molecules that have a bigger gap between their energy levels are going to require a higher energy photon. They're going to require that would be a shorter wavelength, a higher frequency. Often we're typically talking in terms of wavelength, so you just need to remember that the shorter the wavelength, the higher the energy. This is going to come into play because, well, the light that you need to excite you is going to be a higher energy, shorter wavelength light than the light that you're actually going to give off, because it's not totally efficient. So you're going to give off a wavelength that is going to be a longer wavelength, lower energy. So different molecules will have like different electronic floor plans, going back to like our Jablonski diagrams. So for example, like conjugation, so if we have resonance, if we have electron delocalization, that's going to make those gaps smaller. And this is why we can kind of like, if you think about dyes, those are often have a lot of like aromatic rings that can actually reduce the amount of energy needed so much that they're able to absorb visible light. So when they absorb that visible light, then we actually see color because they're stealing part of the rainbow. There's a little bit of wiggle room. So it's not just like quite those distinct states. So you can kind of thinking of being able to like stand on your bed or stand on your dresser and kind of jump up and down between those. But you can't go actually between the floors, but there is a little bit of wiggle room. And this is why for molecules, if you were to go and look at their like absorbance, you were to go and like test all these different wavelengths and see where they absorb more strongly, you'll see that instead of seeing like distinct little peaks, you actually see these like absorbance maxima. So basically you kind of get more of like curvy type stuff. And you'll see you can have multiple peaks because molecules have multiple parts. And remember that, for example, we can take advantage of the different profiles of molecules in order to kind of determine what molecules are present and to actually quantify them. So remember, we saw that the proteins are going to absorb UV light, thanks to the aromatic rings of some of the amino acids, as well as to the peptide backbone. And different molecules absorb different wavelengths of light. So nucleic acid will have a kind of different profile. And again, then we can use the extinction coefficient and Beer's law and all that good stuff to figure out quantities, concentrations. But that's not what we're doing today. Instead, we're going to talk about what happens when you don't just absorb light, but you actually give light back. That is, you fluoresce. In this case, you're going to not just have like one curve, you're going to have an absorption curve and an emission curve. So the absorption curve, remember that this is going to be shorter wavelength, higher energy, higher frequency, higher energy light. So you're going to absorb that higher energy light, and so then you'll have an absorbance peak. Then you'll also have an emission peak. This is the light that you give off. This is going to be a longer wavelength, lower energy light, because remember, lose a little bit in heat, vibrational relaxation, all that non-radiative stuff that's not contributing to the light you give off. So you have less energy in the light that you can give off. And so then you have a lower energy emission. The difference between these two is called the Stokes shift. And the kind of like overlap is going to be called the spectral overlap. And so this is just for a single fluorophore and a single that's absorbing light of one wavelength and emitting light of a different wavelength. This is not the overlap we're going to be seeing between our excitation fluorophore and our acceptor fluorophore. This is just for the single fluorophore. Here's an example of GFP. Remember, we looked at GFP with that classic paper where they figured out that you could actually like use GFP, like genetically encode it, have cells make this GFP, and it was actually functional, and they could use it as a reporter and all this really great stuff. So remember, one of those figures was actually the absorption and emission spectra of GFP. So it's not labeled on this figure, but can you tell which one is going to be the excitation and which one is going to be the emission? Yeah, so remember, our excitation is always going to be the one that has higher energy. And so the excitation is going to be this one. Excitation. So this is our excitation, our absorbance, same idea. Because remember that it's absorbing light and that's exciting the electrons to a higher energy level. So this is going to be our excitation. And then this one, well, this one's going to have a longer wavelength. It's going to be lower energy. This is going to be our emission. So if we were setting up our detector, to detect GFP, basically we would want to have it so that we would be detecting about like 500 or 20 or so, and that we would be exciting it at about 400. So you can see that it kind of has this really broad excitation profile, not really ideal for what you'd want. And it's also absorbing in wavelengths that a bunch of other stuff absorbs in. So they wanted to see if they could make changes that could make it more favorable, have better properties for uses as a fluorophore. Remember that because that GFP has it's fluorophore, like genetically encoded, it's like part of the protein itself. So they could make changes to the protein sequence in order to change that fluorophore. So this is from Chen's Nobel Prize lecture, where he's actually showing some of the mutations that they made in order to try to improve the properties. And so here they're looking at serine 65 and looking and seeing how changing that to a threonine or a cysteine or an alanine affected the excitation profile. You can see that when they made these changes, well here, so in this dotted line, this was the wild type, and here when they made these changes, they were able to shift the excitation curves. Because remember that what light wavelengths they absorb is going to depend on the molecular, like the orbital layout, based on how the different atoms in the molecule are arranged and interacting and all of that stuff. That'll impact that. But you can see that they're all they have like similar emission profiles still, but they are absorbing different wavelengths of light. They did a bunch of further work to try to find even more variants. So basically here, this is this S65T. What they found was they actually got even better emission from their S65T. They were able to get even like stronger light. So they got brighter. They were also able to actually like change the colors by introducing different mutations. And then lots of them have been evolved. So some of them are derived from that original GFP. Others are derived from other fluorescent proteins and evolved in the lab by various strategies, making lots of mutants and seeing what colors they make and if they change colors. And then they get all these fun names like M banana and M honeydew and M tangerine and M plum. So there's a lot of different options for fluorescent proteins. And this was just 2004. Why is it good to have a bunch of different proteins? Well, we talked in class before about how you might want to monitor different things at the same time, maybe track multiple proteins within a cell, or have different cell types light up in different colors. Very cool stuff. Very cool stuff if you have different colors. And you can also have colors that aren't going to interfere with other colors. So having a lot of options is great. But one of the ways that having a lot of options is great is by being able to combine different fluorophores to do FRET experiments. So remember that in FRET, we have that energy transfer from one molecule to another. And then the first molecules, basically, you don't see its light. You're only going to see the light that the second molecule gives off. So it's kind of like you have two apartment buildings, and if their elevators are just lined up right, what can happen is that when one is going down, it kind of hops over to the other next door apartment and takes the ride up to excite that electron. So it's non-radiative transfer. So you're actually, the first one isn't even giving off the light. It's just getting transferred non-radiatively, not through light, to that second molecule. And this can only happen if they're really close together and their energy levels match perfectly, or at least as perfect as allowing for those little like wiggle vibrations and stuff. But you're not going to be hopping from one apartment building to the apartment building 5 blocks over, but maybe you can hop next door. So this is going to allow us to see if molecules are close together. And so, yeah, so actual Jablonski diagrams will look something like this more, but you can see that you have these different energy levels and you get this kind of transfer of energy if you match between if the energies match between the light that's given off by the first one and the light that's absorbed by the second one. And remember that when we talk about the light that's given off or the light that's absorbed. So we're talking about our excitation and our emission, and we're typically we're thinking in terms of wavelength, which is going to be this lambda. So if you have a higher wavelength, remember that a higher wavelength is going to be lower energy. So let's just write that down so we remember. So we have higher wavelength, lower energy. And then we have lower wavelength, higher energy. So our donor is going to get excited. It's going to absorb the higher energy light, the lower wavelength, and then it's going to emit light. And this light that it emits is going to be lower than in energy than it gave off, but it's going to be enough to excite the acceptor. And then the acceptor is going to give off even lower energy of light. Now, the donor and the acceptor, so the emission of the donor has to match the excitation of the acceptor. So you want there to be this like overlap in between them. And there's various things that you want, like you want good brightness, you want good lifetime, and you really want to make sure that your excitation of the first one and your emission of the second one are going to be well separated, so that when you shine light that excites your donor, you're not going to then detect that light. And you also want it so that the light that excites the donor isn't going to overlap with the light that excites the acceptor, or else that's what's going to happen is basically you'd be exciting the acceptor without actually exciting, or even if you didn't excite the donor. So basically, scientists have gone through a lot of trouble to come up with different combinations and different fluorophores, both fluorescent protein as well as chemicals, in order to try to optimize these various properties. A common combination that you might see is like CFP and YFP. So cyan fluorescent protein, and yellow fluorescent protein. And so you'll see basic things like this. So basically in this dotted line, this blue dotted line, or I guess the cyan dotted line, we have the CFP's excitation. And then the solid line, you have its emission. You can then see that its emission is going to overlap with the excitation of YFP. And then YFP is going to give off light of a longer wavelength, lower frequency. So what you're actually dealing with, though, is basically the part that they overlap. So you want the emission of your CFP to overlap with the excitation of your YFP. And so you can see that there is this range in which they overlap. So you can have different combinations of different fluorescent proteins and different small molecules, dyes, and you are not expected to remember all of these. But if you should remember that you need the emission of the first one to be near the excitation of the second one. And so often it won't be like perfect, like because it's talking about like in terms of the maximum, but remember we have that kind of like wiggle room, that more of a curve. But you want to see something where, say, we give off light at 520 and now this one absorbs light at 550. So that's like you have the excitation from the first one is going to excite the second one. And there are a lot of options with a lot of really fun names. And often instead of like just trying to remember which one goes with which, you just look at a table and see which ones normally go together. Okay, so you have this choice of your donor and your acceptor and all of this great stuff. But the real key thing about FRET is that it's distance dependent. If those molecules are close together, then you can get FRET. You can jump from one apartment to another. But if they're far away, they're too far, you can't get that FRET. And so the closer they are, the higher the FRET, and the further they are, the lower the FRET, and to get to a point where you don't have any. So on the this is kind of like our FRET efficiency, so how good that FRET is. And you can see that basically if they're close together, you get this really high FRET, and if they're far apart, you get the low FRET. Now in the middle at this like 50% mark, so this 50% mark, that's going to correspond to your R naught, which is basically going to be the distance between them at which you get half of that FRET efficiency. So this is also sometimes called like the Förster distance, the 50% transfer efficiency. So things get really complicated because it doesn't just depend on distance, it also depends on orientation and a bunch of other stuff like various properties of the fluorophores. Bottom line, you don't need to worry about that. But what I do need you to know is like, yeah, those complications are complicated when you try to like think about the math, but they're actually really great in that they allow us to figure out information about the molecules, not only how close together they are, but also their orientation, their movements. There's a lot of information that we can get just by measuring the fret efficiency. So basically, when we think about frets, one of the most important things is that distance dependence is like really distance dependent. So basically, it's scales to like 1/r⁶, where we basically, the r is going like the distance between the molecules, this r, and then the r naught is basically the property of the fluorophore pair. And so basically, what I just need you to know is that it scales to 1/r⁶. So basically, as you move away, you're going to quickly drop off in fret. And because there's this actual relationship that between them, there's actually a lot of variables that go into these numbers, but people can mathemize those variables and you can actually figure out the r, the distance between these molecules, just by measuring the fret. And then remember this r naught, basically remember that was our 50% transfer efficiency distance. This is typically about 3 nanometers to 7 nanometers. The maximum that you can get and to still see any fret is typically about like 10 nanometers, which remember is 100 angstrom. Okay, and so because you have that mathematical equation, sometimes frets referred to as like a spectroscopic ruler. Because the closer they are together, the higher the fret, the further they are, the lower the fret. And so by measuring the amount of fret, you were then able to determine how close together they are. Now, fluorophores can be like proteins like GFP and YFP and all of those fun fruit names, or they can be small molecules. So this is showing things to scale. This is one of our good friends, our antibodies. And so this is the size of an antibody. You can see this is VFP, it's Verde fluorescent protein, which is a form of GFP. These are small molecule dyes. This is streptavidin, our good friend streptavidin, which remember binds to biotin really, really tight. So basically this is just showing you the scale of different molecules. Here are some examples. So you can have various dyes as well as various proteins, and each of these will have a characteristic absorbance and emission wavelength. In terms of size, so that gave you like a rough idea. Here's a lot of these fluorescent proteins we talk about have these like little beta barrels. And so these beta barrels, remember that would be an example of that's a motif. And so each of these would be beta strands. And then you have this beta sheet that's kind of in this circle. And it looks kind of like a channel, but there's actually a bunch of side chains sticking in here so that the water can't get in. And this fluorophores is nice and protected, and it's able to do its shining. But it's also going to be big. And so we're already taking like 2 nanometers or so from our distance that we have. And remember, we only had about 5 nanometers. And so now we're saying, okay, well, like 2 of the nanometers is actually taken up by the protein itself. So that's one of the downsides about protein sensors, is that they're kind of big. And not only does that make it harder to see for like the molecules have to get closer in order to see them, but it also means that you could have, if you put a big molecule onto your protein, that might actually affect how your protein is functioning. Small molecules, here's an example, Psi3, this is a common one. You also see things like Psi5, not Psi5, Psi5, and basically, so this is Psi3, it is maximally excited at 550 nanometers and it's emissions at 570 nanometers. So if you were to go and want to use Psi3 as a fret donor, so that first partner in the pair, what would you want to use as your acceptor? Not a specific name, but what would you want its absorbance to be? Yeah, you'd want it to be around 570 nanometers because that's what this Psi3 is going to give off. So why don't you pause the video and think a little bit about some of the benefits and drawbacks of both small molecules and fluorescent proteins. Okay, so basically with small molecules, well, they're small. So there's less risk of interfering. Can detect longer distances. Between molecules. Because remember, you don't have, it's not that the fluorophore itself is that, but it's basically because you don't have that bigger, you don't have to consider the size of the protein taking up some of that distance. And so fluorophores can be really great. They're more stable, but they also have downsides. So let's think about what are some of the benefits of having a fluorescent protein. Probably the main benefit is that you can be genetically encoded. So basically, we can attach a GFP, we can attach a YFP, we can attach a CFP onto the ends of proteins. So we can attach it to like the N terminus, or we can attach it to the C terminus, just by sticking the sequence for that protein onto the sequence of our protein by making one of those fusion proteins. So just like in our lab, we had that GST fusion protein, you can also make a fusion protein with one of these fluorescent proteins. Now, you can't genetically encode the instructions for putting on site 3 or putting on one of those small molecules, at least within the cell, but you can genetically encode the instructions for making that fluorescent protein. So that is one of the main benefits of having a fluorescent protein, is being able to get cells to attach it to proteins when those proteins are actually made inside of the cells. We'll look at some examples later in which they actually like genetically encode sequences that can then be modified to add a fluorophore, like to conjugate a fluorophore onto those direct sequences. But that can't be done that's not done like inside of cells. That's if you're working more like in vitro. Whereas if we do this inside of cells, well, now we're able to monitor what's going on inside of cells and even maybe inside of organisms. So that genetic encoding of those fluorescent proteins is really, really going to be a huge benefit. But those small molecules are really helpful too, and I don't have time to go into it, but if you're interested, there are basically various small molecules that can be used as FRET sensors, that they either like change their fluorescence properties or they get modified in some way in the presence of various chemicals or various stimuli, and this is then going to change the FRET that they participate in, and can therefore be used to detect things like the amounts of metals and the amounts of various chemicals and light and all of this really cool stuff. But let's talk more about kind of just our typical NE-FRET. And so what can happen is that you can have different arrangements of your donor and of your acceptor in order to detect different things. And so there are lots and lots of different ways that FRET can be used as biosensors, so sensing what's going on in some biological context. And here are a few strategies. So basically what you can do is if you can have like intermolecular, so between molecules, or intramolecular within the same molecule. So in A, we have an example of an intermolecular FRET. So we have different fluorophore. We have a fluorophore on 2 different molecules. So we have our donor fluorophore on one molecule and our acceptor fluorophore on another molecule. This could be 2 proteins, or it could be a protein and a small molecule, maybe like a drug or something. But basically what's going to happen is that only when they come together, only when they bind, are you able to then get FRET. So if they're not bound and you excite the first you shine light that excites the first fluorophore, then you're going to see the emission of the first fluorophore. But when they bind, well, now if you excite that light that excites the first fluorophore, you're going to see the emission of the second fluorophore. Here's another example. This one is intramolecular. So I really need you to know your prefixes. So this is intermolecular, inter between, and intramolecular, intra within. So basically here you have your donor and acceptor are on the same molecule, they're on like the same protein, but on different parts. And so what's going to happen is that then if the protein undergoes a conformational change, then you can detect that. So this would be an example of a conformational change. And in this case, they're showing it being a conformational change that is kind of caused by the binding of a ligand. But you can also detect conformational changes that are just due to like molecular movements for other reasons. So what you can do is you can there are various strategies, but you might have one on the N terminus and one on the C terminus if you're doing like genetically encoded fusion proteins. So you could say have like CFP on one end and then like YFP on your other end or something like this. There are also ways you can actually, if there's a flexible domain in between them, you can put like a fluorophore, the instructions for a fluorophore in kind of like the middle of the protein. Or as we'll see, there are various strategies where you can use a short sequence that then will get chemically modified. So lots of different strategies, but basically you need to make sure that you're introducing the fluorophores in a place where they're actually going to be close enough to interact at some point. There are also ways where you can kind of test for modifications, things like post-translational modifications, phosphorylation. You can have a substrate that's attached to a sensory domain. So basically what's going to happen is that the substrate, so this like amino acid sequence, if it gets phosphorylated, say, what's going to happen is then it's going to bind to this domain of this protein that actually recognizes phosphorylated sites. We'll look at an example in a second, so hopefully this will make a little more sense. But basically what's going to happen then is that this is going to bind, this is going to pull the acceptor fluorophore close by, and now you're going to get fret. You can kind of have the loss of fret as well. So these have all been examples of like fret on, but you can also have fret off, where basically if you have you have your two fluorophores attached together, and then say a protease, so a protein cutter cuts between them, they're going to get separated and now you lose your fret. You can also have similar things where you're looking for a change in shape based on the loss of fret. So you can either monitor the gain of fret or the loss of fret. In either case, you're able to tell information about the relative locations and potentially modifications, depending on how you set up your fret strategy. You can also do cool things to use fret to kind of like measure forces and based on distances. And you can do things like, this isn't fret, but you can actually have like split GFP where you have two parts of the fluorescent protein, like the protein made in kind of like halves that then have to come together. Various really cool strategies for using fluorescence and using fret as biosensors. One example is basically what you can do is you can have antibodies that are labeled with fluorophores. And this way you can have, if you have two antibodies that recognize something, such as two different antibodies that recognize one of the COVID proteins, this is like the nucleocapsid protein, NP, what's going to happen is basically one of the antibodies bind to, when you have two antibodies with one's a donor and one's an acceptor, bind to that NP protein, they're going to be close enough together that you're going to get fret. And so this can be used to detect the presence of the virus, as well as then to screen for things that prevent the virus from infecting cells. So very cool strategies that you can use, lots of strategies that you can use for fret. So this is an example with using antibodies as a way to prevent having to actually label your protein itself. There are also all sorts of strategies where instead of just having one donor and one acceptor, you can have like multiple colors and you can have multiple pairs and all sorts of really complicated schemes that I'm not going to get into. What I do want to mention though is like sometimes we can do fret inside of cells, and sometimes what we're doing is we're actually doing it on like immobilized molecules and like single molecule fret. So this is actually a cool example where they're trying to measure not just are things interacting or where are things interacting. So remember, if you're in a cell, because you can have the fluorescence, you only get that fret in the specific area where molecules are interacting. So you can actually use this to localize things within cells. But you can also use like fancy methods in order to try to study like dynamics and kinetics actually inside of cells. So in this example, what they're doing is they're actually like micro injecting cells with one of the proteins. So they have one of the proteins is going to be expressed in the cell with a fluorescent protein fused to it. And then you have a second protein that's going to be injected into the cell. Now what's going to happen is they can start monitoring. So in the top panel, so this is going to be 0 seconds. So basically before they inject, then this is going to be them after they're injecting. So this is going to be 1 second. And this is going to be 30 seconds. So in the first panel, they're showing the emission of the donor. And so remember, you excite the donor and then it emits, and you can measure that. And in the second one, we're monitoring FRET. So this would be looking at the excitation of the acceptor. So this is the excitation of the donor and the excitation of the acceptor. What would you expect to see if the two molecules are interacting? Yeah, you'd expect to see a decrease in the emission of your donor and an increase in the emission of your acceptor. And you can see that that's actually what's happening over time, is that as you start interacting, you're getting a decrease in this in the donor channel and an increase in your FRET channel. And then this is just showing you kind of like what things look like in the absence of fluorescence. And I think they're showing the tip of their probe, which is kind of cool. I actually did something similar with like it's like nanobiopsy type thing where I was actually trying to take things out of cells. Didn't work so well, but it was a great learning experience. Those are all examples of kind of like how you could do things inside of cells. And so remember, inside of cells, we can localize things. We can study what's going on in like a real environment. But often, as we talked about the last week, we want to do things kind of with where we can control things better. We want to really get down to the nitty gritties. We need more control. And inside of cells, it's hard to tell, like, what molecules, like, are we actually dealing with? How many? You don't get the kind of resolution that you might want. Another option is single molecule FRET or SM FRET. Typically, you have things immobilized. You don't always have things immobilized, but you can have like a slide. And this is often, you then attach a molecule to the slide, and that molecule is going to be labeled with the fluorophores. Or you can have one molecule that's labeled and then you flow other things over it to see if they interact. But basically what's going to happen is somehow you're attached to a chip. You're attached to a slide in this case. And this is going to be used often in conjunction with internal reflection spectroscopy or TIRF. Basically what's going to happen here is that you have your samples on a slide, and then you shine the light and detect the light. And there's all sorts of fancy dancy spectroscopy and microscopy stuff going into it that I'm not going to deal with. But often you're going to have things like immobilized with biotin and streptavidin. These are going to be really common. Various strategies you can use, like here they have a nickel on here and then a his tag on the protein. So remember, we talked about how we could use like nickel resin to purify proteins that have a his tag. We can also use like nickel coated slides in order to capture his tagged proteins. And then if these have the fluorophores on them, well, now you have a way where you have these single molecules immobilized, and you can then study how they are moving and interacting in this sort of things at a much greater resolution than you would be able to inside of cells. Let's look at an example of what some of this data might actually look like when in one of these single molecule experiments. So here they have this molecule with a donor on one end and an acceptor on the other end. And it's linked to this slide through biotin and streptavidin. What's going to happen is that when the molecule is in a conformation in which the donor and acceptor are close together, you're going to see FRET. And when they come apart, you're going to lose that FRET. Now, these molecules might be going back and forth and back and forth and back and forth. So you can actually measure their conformational dynamics. You can measure the switching from the FRET to the high FRET state to a low FRET state to a high FRET state, and see something like this, where you have on your y-axis, you have like the FRET. So we can think about our FRET efficiency, remember, is going to be dependent on that distance. And then we can basically graph the amount of fret versus time in order to get an idea about how much time these molecules are spending in these different conformations. What you might also see is like a direct counts. So basically what they do is they take the counts. So basically how much fluorescence do they see from their donor wave, from the donor emission wave like channel or from the acceptor. And so let's look, and then what they can do is they can like graph the ratio of those, and then they get this like fret signal that like we were looking at before. But what they originally get is something like this. So you might see something with counts. So in this example, basically what they have is that this green trace is going to correspond to the emission of the donor. And then the red is going to correspond to the emission of the acceptor. What's going to happen is that if they were not interacting, what you would see was that you would basically not see the acceptor fluorescence, but you would only see the donor fluorescence. And so when you have something like this, and then you look down, this is going to show you that you don't have fret. Fret's not going on here. Don't worry about photo blinking or photo bleaching. Those are just like what happens when the fluorophores get tired or do weird stuff. But what you see when you have basically this really, really low amount of your acceptor emission and a really high amount of your donor emission, this is going to be an instance where you do not have fret. So these are examples of places where you don't have fret. So the molecules are far away. But then you have examples where they come together. And so what is characteristic of fret is that you're going to see a decrease in the donor at the same time you see an increase in the acceptor. And so you're going to get something like this. And this then is going to correspond to high fret. Note that in the last example, it was really nice. It was like we're going from like 0 to 1 or whatever. But things are typically more messy, which can be annoying, but it can also be helpful because not only are we seeing like these different conformational changes, but we can actually detect that they're actually kind of different energy level states. So there's actually like, or different efficiency states. So they're kind of like, remember, because it has that distance dependency, if they're kind of an intermediate amount apart, you can actually detect that. So fret can be used to detect like different states of different conformations. And then often what's going to happen is that they'll plot it on some sort of like histogram, which shows like the probability that it's in one state versus the other. So there are different ways that you'll see fret data, and we'll look at some examples and things like this to help solidify the concept and so that we can understand what's going on. So let's look at a couple examples that are both going to involve that spike protein. So remember from our structural biology lesson, that spike protein was the coronavirus protein that sticks up off of the surface of the virus and allows it to latch on to cells. And it does that by binding to the ACE2 receptor. So ACE2 is a membrane protein in our cells that binds to the spike protein in the viral cells. And then this allows for a shape change that's going to then bring merge the virus with our cells and allow the virus to dump its stuff in. So in one of the strategies for kind of combating the virus would be to prevent the binding of the spike protein to ACE2. And remember that the specific region that binds is this receptor binding domain. So what they're going to do is they're going to use fret to measure the binding of the receptor binding domain to ACE2 in living cells. So TR, this is like time resolved. Don't worry about it. It's just a type of fret. So what are they going to do in this experiment? Well, if they want to measure the interaction between the RBD and the ACE2 protein, what do they need to do? Yeah, they need to label the two of them. And they're going to kind of take different strategies for ACE2 and for the RBD. And so for ACE2, they use this thing called a snap tag. Basically, it's this little enzyme that you can fuse with your protein. So instead of directly fusing a fluorescent protein, you can fuse this enzyme, which will react with a fluorescent molecule that you add or other types of molecules, form a bond to it, and now you have get it labeled this way. And there are various pros and cons to strategies, but they put a snap tag on the end of ACE2. So this is going to then allow them to label the ACE2 protein. In order to label D2, well, here basically what they did was they used lysine labeling. So lysine, remember what functional group do we have with lysine? Yeah, we have an amine group, and the amine group is amenable to modifications. And so basically you can kind of conjugate it with this D2 dye. So they're basically able to use that lysine to label the lysines with a dye. And this is a common strategy that's used to label various proteins, but it's not going to be as sequence specific. There are also ways that you can label cysteine. Again, these aren't details that you need to know. Just know that there are ways that you can fluorescently label proteins if you're working with them in vitro. So because they're basically going to be purifying this RBD, so the receptor binding domain, they can then modify it and then do their experiments. Also, they kind of have this unique situation with ACE2 because it's a membrane protein where they can basically do this reaction while the protein's embedded in the membrane. So cool strategy so that you can actually be dealing with living cells, but still modifying the protein. So now what they want to do is they want to detect the interaction and the loss of interaction if you have an inhibitor present. So we have a fluorophore on our ACE2, which is called this Lumi4TB. And then we have a fluorophore on our receptor binding domain, our D2. What's going to happen is that if the receptor binding domain binds to ACE2, so binds to our cellular receptor, we're going to get FRET. So if we shine light that excites this Lumi4TB, we should see the emission of our D2. But if they're not, only if they're interacting. If they're not, then if we shine light that excites this terbium, this Lumi4TB, what's going to happen is we'll see the emission of that. And so this is what they end up doing. And so here they're doing like a binding assay with this, and so we should be very familiar with our binding assays by now. And so what kind of graphs are we looking at here? Yeah, so here we're looking at one of those like equilibrium binding graphs. We're looking at concentration over concentration of our x-axis, and then they have this like FRET ratio on our y-axis. So the more FRET you have, the higher the value here. And what would give you more FRET? Is that indicating binding or not binding? Yeah, in this case, it's indicating binding. So they're showing that when you get binding, you get this higher FRET. They're measuring like total binding and like non-specific and stuff. Basically, they have a control to test for non-specific binding, where they have a bunch of unlabeled stuff to see if a bunch of unlabeled RBD to see if they're still seeing fluorescence. And so they're seeing they don't get fluorescence if they've saturated their receptor with the RBD and all's good. And so then they say, okay, well, that's all basically, if you subtract that, we can consider that all this binding we're seeing is basically specific binding. Okay, so from a graph like this, we can go and we can determine the KD, right? And so what would our KD be in this case, approximately? Yeah, it's going to be about 15 or so. Basically, it looks like the peak is about like 2,500, 3,000 or so. Go somewhere over here, come down. Yeah, you're about 15 nanomolar or so. And so this is like one of the reasons why it's kind of nice to have those log graphs, right? Because you can actually see the inflection point better, and these points aren't all scrunched up. But anyway, this is what we have, and you can determine things like KD. Can we tell anything about the kinetics here? Can we tell about K on and K off? No, we can't. We can if we only have the KD, if we only have this equilibrium data, we can't tell about kinetics. But with their strategy, well, they are able to tell about kinetics. And so they can set up their experiments. So what's going to happen is that they can measure the actual binding. And so when you get this, you can see association. You can get your K on. And then if they then do a dissociation assay, they're then able to see the release over time and get the K off. And so that's what they're showing in this figure. And note that here they're basically showing how they got the KD from the kinetic data. versus how they got the KD from the equilibrium data. And so you can see that they're not exactly the same, but they're within like a few factors of one another. Okay, so now I want to go and they want to look and use this as a screen for inhibitors. So if an inhibitor is present, do we expect to see more fret or less fret? We would expect to see less fret because you're not going to have as much of the interaction there. So if you were to mix competitor and your labeled RBD, well, now what's going to happen is that the more competitor you have, the less of the RBD that's going to be the labeled RBD that will be bound. In this first case, so here they have basically unlabeled RBD. So remember, that's basically like our reference point. So we have the labeled RBD and we know that, so we're measuring the amount of labeled RBD bound, but we need to have some way to know what the actual affinity of the RBD itself is, as well as to make sure that that label doesn't mess things up. And so that's what they have here, is we're seeing the competition binding curve for the unlabeled RBD compared to like the labeled RBD. And so in here we have this LCB1B3, which is this little protein like peptide thing that they engineer to try to bind and block it. So by looking at this curve, so which is going to be the stronger competitor, which is going to be the stronger binder to that ACE2? Yeah, it's going to be this LCB1B3 because we see that it's shifted to the left. And so we can actually kind of get a rough idea about our KIs for the molecules. So what's our KI for this LCB1B3? And how do we know? Well, we want to look kind of like the halfway point. And you can see that here they have a log scale, so things are a lot easier to see. And so it looks like we're dealing with something about here, somewhere on the order of like 10 to the minus 9, 10 to the minus 10. So basically a little under like sub-nanomolar range. Whereas our like K apparent for our RBD, this is going to be more in the -7 to -8 range. So remember that the lower the KD, the higher the affinity, the lower the KI, the higher the affinity. And so basically this, and it will be shifted to the left. This is going to be the stronger binder. This is another example. This was just kind of cool. They did this like nanobody. Basically, llamas have these really cool, like tiny little antibodies, like these that you can make these like nanobodies, which are then smaller, easier to get into cells and things, and kind of can wiggle into nooks and crannies. So that's the strategy that some people use to target all sorts of things. And so they were testing the nanobody binding, and that's what's shown here. Okay, so that was measuring binding. So there we had two molecules that was intermolecular. We had a fluorophore on one molecule that excited a fluorophore on another molecule. Now let's look at an example of intramolecular fret, where we're looking at, we have two fluorophores on the same molecule. In this case, we're going to use it to study conformational dynamics. So that spike protein, remember, it's the RBD that's going to be binding to ACE2. And in order to do this, it kind of has to adopt this up conformation. So the spike protein, basically, it's a homotrimer. So it has these 3 copies of the same protein. And basically, each of those copies is going to have one of these receptor binding domains. And it can be in the down conformation or it can be in the up conformation. And when it's in the up conformation, this is when it can bind to ACE2. And so scientists wanted to study how often it goes up and down and up and down and up and down and up and down. And so in order to do this, they were going to use fret. So basically, because they're going to take advantage of the fact that if fluorophores are close together, they'll be able to see fret. And if they're far apart, they'll be able to they won't see fret. So by designing these, fluorophores like locations strategically, you can then monitor conformational changes in a protein. So in this paper, that's what they did, and they wanted to see how specific variants of the protein affect things. So how do mutations that change the protein affect its or do they affect how often it's going up and down and up and down and all that stuff? So this is a really cool paper by Yang et al. That I thought I would use as an example. What they're going to be doing is they basically have these like lentivirus particles. So basically this is like a safe virus, but they put the instructions for making the spike protein. And they can then anchor this virus onto a slide. So basically they use this like biotinylated lipid so that they can then attach it to streptavidin, which is attached to biotin on the slide. So they have a way to immobilize this virion on the slide surface, so that they're able to measure things kind of like in a more natural environment, but still have that tight control. In order to be able to measure the dynamics, they're going to introduce 2 fluorophores. So how they're going to do this is basically, so in here, this red dot is going to be this like A4, this is one of their fluorophores, and this green dot is going to be this Q3, another one of their fluorophores. And so you can see that this is when all of them are down, are these close together or far apart? Compared to over here, where one of them is open. Yeah, these are going to be closer together here. So are you going to see more fret if they're all down, or more fret if one is up? Yeah, you're going to see more fret if they're down. So you'll get higher fret if you are in the down conformation. Now, because you have that tetramer, things can get really complicated if you were to have like all of them labeled. So they basically were very strategic and only labeled like a small amount of them and had most of it be unlabeled, so that they would on average have like one subunit that had both labels per spike protein. And therefore they would have the least amount of noise. As to how they actually did that labeling, it's pretty cool. And they kind of like put these really, really short little sequences inside of the protein sequence. So instead of putting them at the end, that wouldn't work for them because, well, the protein, they'd be like way too far apart. You wouldn't be able to see them. See if you had fret or not. We need to choose sites that are going to be close together, but then how are we going to get them labeled? To do this, basically what they did was they used these cool little like sequences that are recognized by this specific protein, by these specific enzymes. So by like ACPS, as well as like transglutaminase and stuff. Basically, they have ways that they can then introduce specific labels in specific locations, and then by modifying, like by being careful about how much they use, they're then able to get on average one of the spike protomers, so one of the individual units labeled per spike protein. And this is going to then allow them to study the dynamics. And so what you see is you get something like this. So remember that this is kind of good. So this is our like fluorescence. This is basically our counts. So you can measure the fluorescence of the donor and the fluorescence of the acceptor. So if you have a region like this whole region, like these regions, what is this showing us compared to this? Well, in this region, what we see is basically we're seeing mainly the emission of our donor and very little of our acceptor. So we're not seeing fret here. And indeed, if we look down, you can see that there's not fret. But if we look at somewhere place like here, well, here we see the decrease in the donor, an increase in the acceptor. And if you look down here, yeah, we do have fret occurring. And you can see that kind of like we saw before, you can have intermediate levels of fret. So they talk about like open versus closed versus partially open. So they see these different populations. So what do you think would correspond to the open conformation up here? So remember that when they're fully open, those fluorophores are going to be far apart from one another. And so you're not going to see fret. And so the open would correspond to things happening down here. What would correspond to the closed? Yeah, the closed would be up here where you see that high fret because they're going to be close together. So what they wanted to do is basically look and see if the mutations were going to influence things. It's kind of confusing because basically D614, this is going to be the wild type. So this is our wild type, and this is D614G, but they write it G614, which is weird, but basically this is going to be our mutant version. This is one of the earliest mutations that occurred and kind of really quickly took over. And so scientists were trying to figure out why. And one of the reasons they thought was that it might influence how that receptor binding is going up, down, because you can imagine if it needs to be in the up to bind to ACE2, if you had a mutation that made it up more often, this would increase its probability of binding and like being able to infect cells. And so what they see was that if we go and we look at that wild type, we can see that it spends a lot of time in each of these different states. It spends a lot of time in the closed state, not that much in the opened and not that much in this partially open. But what happens with the mutation is now you're spending more time in this partially open state. And this can make it so that it's then able to bind ACE2 more readily. What happens if you actually add ACE2? Well, if you add ACE2, well, now what's going to happen is you're going to stabilize that open state. And in fact, that's what they see. So both with the wild type and with their mutants, if they add ACE2, they see that they're stabilizing this wild type. So they're stabilizing the open conformation. So again, does that open conformation correspond to low fret or high fret? Yeah, that's going to correspond to low fret. So that would mean that you would have more of these instances where you would have things like this, where you're seeing mainly the excitation of the donor and not the acceptor. So I know that was a lot, but hopefully the examples were helpful and kind of cool and not just overwhelming. Remember that you don't need to know all those details. They were just examples. And instead, what I want you to focus on is those concepts. So with fret, you have two molecules, two fluorophores. The first fluorophore absorbs light of one wavelength, and it emits light of a wavelength that excites the second fluorophore. And then the second fluorophore is going to emit light. Remember that each time you're emitting light, it's going to be a lower energy, longer wavelength than the light that you absorbed, because nothing is totally efficient and you lose some through heat and vibration and all that stuff. So you're going to start seeing lower energy light if you get fret. In order to get fret, well, these things have to be close together. Close enough together that the energy can actually kind of just jump from the first thing to the second thing without light of the first thing even being given off. And so that's what fret is this non-radiative or non-light energy transfer. It can only happen if they're really, really close, within like 10 nanometers at the greatest distance. But this and the actual distance, like it drops off at an order of like the radius of the sixth. So it quickly drops off. But it drops off at a rate at which we can then actually use the amount of fret to figure out how close together molecules are, or how close together different parts of the same molecule are. So we can use it to measure both intermolecular between molecules and intramolecular within a molecule dynamics and things. You also have quench fluorescence, where basically what would have been your second fluorophore is now a quencher, which basically just steals that energy, doesn't allow the first molecule to give off light, but also doesn't give off light itself. And there are various methods that we use, all of these different things. We can do things inside of cells, including with like genetic encoded sensors. So using things like GFP and YFP and all those FPs, through those recombination, molecular cloning and all that good stuff. Or we can use small molecules. And we can do things inside of cells, or we can do things like immobilized on chips with like single molecule fret. So lots of really cool stuff. And we'll take a look at some more examples and get a little more practice with interpreting the data and kind of thinking about how to set up various fret experiments, how to design things to test different hypotheses.